And also, this this nutrients excess nutrients can also affect corals through increasing the prevalence of coral reef diseases. And so, as I said, I'll be talking more about the nitrogen portion. And so, excess nutrients can come from land-based pollutants such as soils, sewage, fertilizers, etc. And they all contain nitrogen within them. And using stable isotopes, we can identify certain sources that are going into the water through certain ranges that have been established through other studies. So how are these nutrients getting to the waters or being transported to the waters? In areas where there's not major rivers or deltas, there's something called submarine groundwater discharge. And submarine groundwater discharge is groundwater that's released at the coastlines through shoreline springs, which you can see in the picture on the right. It's a picture of the fresh water bubbling up right at that shoreline, or through benthic seeps. So if you're swimming and you're snorkeling and you see this kind of cloudiness, this could be a benthic seep. And this is because the fresh water is going into the marine water, so you can see the mixing of the waters right there. So as I said, S2D is fresh water, so it does mix both onshore, offshore, as well as going from the benthic and rising to the surface because it is fresh water. And they can impact coral reef health because with that groundwater comes a lot of nutrients, which can become in excess once it reaches these coastal waters. And these nutrients, as I said, could affect and create eutrophication where the algae grows and can impair coral health. So for my study, I had two objectives. I wanted to identify sources of nutrient pollution in these coastal waters that were fronting to coastal developments. And then my second objective was to document the spatial distribution of nutrient concentrations within the waters. So again, both onshore, offshore, as well as surface and benthic. So my research took place on the leeward side of Hawaii Islands, and my sites were located within the NOAA Habitat Focus Area. And this is just an area of resource management concern, where one of the objectives was to improve coral health by reducing land-based pollutants such as sediments and nutrients. So my first study site, this red dot located within the blue habitat focus area, is Pauoa, located within the Waikoloa watershed. And then this purple dot here is Kukio, which is located within the Kukio watershed. And both of these are areas with developments. They have coral reefs right in front of the developments. And land cover for both of these areas within 5 kilometers is about 2% developed, made up of luxury resorts, residential areas, and a lot of golf courses as well. And in addition to that 2%, about 60% of it is about 60% of the 5 kilometer radius is bare substrate or lava. And both of these developments had strong resource manager and community interests, and that's why these sites were chosen. So my study design, looking at both of these watersheds, we chose 4 different water types from the watershed. So on the left map here, we have Pauoa. On the right map, we have Kukio. And we have starting from high elevation groundwater wells, anchoring pond stations, shoreline stations, and surface and benthic stations. So the groundwater wells are the stars in this top right inset box here. We have the pond sites, which are these 2 diamonds here. The pentagons along the shore are the shoreline stations, and then the circles are for surface and benthic waters. And then we also had fertilized soils and wastewater treatment plant samples, as seen in the top right inset box. So the fertilized soils were from the developments here, and then the wastewater treatment plants were a little bit above the developments here. And the soils and the wastewater treatment samples were used as nitrogen sources to be compared to from the water samples taken from each site. So for my first objective, it was to identify sources of nutrient pollution in these coastal waters that were right in front of coastal developments. And previous research has found SGD to be a contributing source of nutrients to marine waters. Nee et al. 2010 found that development can increase nutrient concentrations, and SGD can be a conduit of that and impair water quality as well as coral reef health. And on Maui, where they have other nutrient pollution, it was found that SGD can contribute up to 50 times greater nutrient concentrations than ambient seawater, and therefore decreasing calcifications of corals. So how we approached this in this study is we had 25 stations total that were sampled 2 times. So starting from the high elevation groundwater wells, we had 2 to 3 of those stations, 2 ancholine pond stations, 5 shoreline stations, and 15 surface and benthic stations. And water samples were collected at all of these. And so what we measured with these water samples were salinity, temperature, nitrate, nitrite, phosphate, ammonium, silica, and then also stable isotope nitrogen 15 and oxygen 18 in nitrate. And that was used for the stable isotope mixing models, which I'll mention in a second. We also used the nutrient data to characterize nutrient behavior by using mixing plots. So looking at nutrients versus salinity from that gradient of freshwater to saltwater. And we also collected algal samples at the shoreline and the benthos, and we analyzed those for the nitrogen 15 isotope. Algae can be used as a proxy for some pollution studies. It's widely established for fertilizers and sewage. And then we also collected soils and wastewater effluent for nitrogen 15 in nitrate to use to compare that data as a source value. And we use the nitrogen 15 in nitrate as well to use in mixing models, stable isotope mixing models, Bayesian mixing models, and use that to further identify sources and find percent contributions. So to analyze the nutrient behavior, we created mixing plots. So this is looking at salinity and nutrients and connecting the freshwater samples to the saltwater samples. So Pauoa here on these figures are on the left, Kukio is on the right, and then we have salinity on the x-axis and the nutrient of interest on the y-axis. So we have nitrate and silica in moles per liter, micromoles per liter. And so the stations or the data that falls along this mixing line, right here is where the freshwater samples are, and then here is the saltwater samples. And then down here characterizes the station types. So this is just a theoretical line, assuming that there's natural dilution from freshwater to saltwater. And so we see that in this top left figure for Paoa, for nitrate, we have the pond stations and shoreline stations above the mixing line. And this can indicate just like there's a mechanism of addition to the system. And then conversely, we can see that at Kukio for silica, a lot of the stations, again, the pond, mostly the pond, Ancholine Pond and the shoreline stations fall below the theoretical mixing line, which indicates that there is a removal mechanism. So at both developments, there were removals. We saw evidence of removals for phosphate and silica. And then we saw additions where some of the stations fell above that mixing line for nitrate nitrite. So the second part of my first objective was to use stable isotopes, specifically nitrogen-15, in algae to characterize nitrogen sources, because nitrogen sources have those specific ranges for certain sources such as sewage and fertilizers. And algae can be used as a bioindicator because they're uptaking this nitrogen and they don't discriminate. So these maps here, we have Paoa on the left and Kukio on the right. And these are showing the nitrogen-15 values from the macroalgae for shoreline here, and then we have the benthic stations here. And so the cooler colors are lower nitrogen or less positive, and then the warmer colors are more positive with more nitrogen. And so what we saw was that the shoreline values were high at both developments, and that the offshore values were lower, and that could be indicative of ocean water or nitrogen fixation decreasing those values. So next we looked at trying to put it more into context of what those values mean against these nitrogen sources. So we plotted these against the nitrate sources that we took from in situ samples, as well as the fertilized soils and those wastewater treatment samples. So the figures on the right, there's Paoa on the top and then Kukio on the bottom. We have the stations on the x-axis and then the nitrogen values on the y-axis. And then the triangles are shoreline samples and the circles are benthic. And then these boxes here show the range values for these different types of nitrogen sources. And so both of the developments fell within ocean and high elevation groundwater ranges, where Kukio did have an overlapping source for fertilized soils. The nitrogen sources were site specific, and so there was a greater range for fertilized soils for Kukio versus Paoa. So Kukio's stations also fell within not only ocean and high elevation groundwater, but also fell within fertilized soils. And then although stable isotope nitrogen-15 in macroalgal tissue is established for other pollutant sources, values can overlap, and a location that has multiple nitrogen sources, it can be hard to distinguish or determine the percent contribution of those sources. So therefore, in addition to the nitrogen-15 in the macroalgal tissue, we use the nitrogen-15 and oxygen-18 in nitrate in these Bayesian mixing models. So shown here on the right is the mixing model output. So On the left panels here are Pauoa, the right panels here are Kukio. On the x-axis, we have high elevation groundwater, soil, sewage, and ocean, so just the different sources. And then we have the proportions from 0 to 1 on the y-axis. And then the top two panels, so C and G, are the surface, for the surface water types, and then D and H, that's for the benthic water types. And then these are your basic box and whisker plots. So the vertical line shows the min and max, or the range of the source values, or the water type values for each source. And then the horizontal line shows that median for each source. So overall, we found that high elevation groundwater was contributing, was the greater nitrogen contributor to all water types. And this was at a 50% credible interval, which contributed, across all the water types, contributed about 52 to 95% of high elevation groundwater. And at Kukio, we found that fertilized soils was the main nitrogen contributor to these coastal waters. And again, this was at a 50% credible interval with 31 to 51% contribution across all water types. And so the nitrogen 15 in the algae, and also with the mixing models, using the data, the in situ data with the mixing models, they concurred with each other and just supported that Pauoa was mainly high elevation groundwater, and Kukio was mainly, nitrogen source was mainly fertilized soils. So again, my first objective was identifying the sources to these coastal waters. And what we found was that both developments had conservative and non-conservative nutrient behaviors. So they had additions and removals of certain nutrients, and that the nitrogen 15 in macroalgal tissue suggests that there are natural and anthropogenic nitrogen sources, and that using the nitrogen 15 in the macroalgal tissue, and then also using it in the stable isotope mixing models, they were able to support each other, and we found that the same sources were contributing. So for my second objective, it was to document the spatial distribution of these nutrients. So looking at onshore, offshore, surface, and benthic. And previous studies have really focused on looking at the shoreline of SGD and some surface, but there's few that has really focused on the benthos of around coral reefs. And this study really incorporated long-term salinity and water data for looking at SGD and nutrients. So how we approached this is we had the in situ water samples collected from objective 1. So when we collected from those 25 stations twice, looking at salinity, temperature, nitrate, nitrite, phosphate, ammonium, silica, and then also the isotopes, nitrogen and oxygen in nitrate. At the same time, we deployed temperature and conductivity sensors that were deployed on the benthos for about 55 days. So here is a picture of one of the loggers that was tied to the benthos, and it's near some coral. And these recorded every 5 minutes for 55 days. So from these in situ measurements, we were able to see a snapshot of where SGD might be, where possible areas of SGD might be. So these maps show the average salinities at both developments. And we have here in the, the left is Pauoa, the right is Kukio, and then this is showing the groundwater, ancholine ponds, shoreline, and surface and benthic stations. So the bottom right inset is the groundwater wells, and then here we have the shoreline and ancholine ponds, and then the circles within the squares are the surface stations, and then the squares are the benthic stations. And then the cooler colors indicate lower salinities, and then the warmer colors indicate higher salinities. And these groundwater wells, again, are just within the watershed where these study sites were. So we found that the shoreline values were lower, and this is indicative of groundwater inputs ranging from like about 0 to 25.2. And then we can see that in this circle here, these orange circles within the squares are surface stations, and those are lower salinities than the warmer color, the red. So that ranged from about 25.2 to 33. And that also supports that there is SGD that could be floating to the surface, creating that density difference and lower salinity measurement. And Kukio did not have the same kind of patterns. There was more uniform offshore, ranging from about 33 to 35. And yeah, so we know that some of the surface stations had lower salinities, and then as well as one of the benthic stations at Pauoa as well had a lower salinity. Next, we looked at nitrates from these in situ samples. Again, following a similar pattern to the lower salinities, where some of the stations with lower salinities had higher concentrations of nutrients or nitrates. And same layout as before, we have Pauoa on the left, Kukio on the right, and then we have the groundwater wells in the bottom right inset maps here for each watershed, and then we have the shoreline and ancholine pond stations here, and then the offshore stations with the circles being the surface stations, and then the squares being the benthic. And the cooler colors are lower concentrations, with the warmer colors being greater in concentrations. And so the purple here is about 0 to 11.5 micromoles per liter. And then these pink ones are about 11.5 to 32.72 micromoles per liter. So that's quite a difference in scale between the assumed ambient benthic and surface nitrate concentrations. And same for Kukio over here, we have 2 surface stations between that range as well. But there were more stations at Pauoa that did have higher nitrate concentrations than Kukio. But overall, nitrate was found to be higher at the wells, the shoreline, and also some of the surface waters. So again, using the in situ measurements, we looked at benthic salinities and benthic nitrate to see if there was a relationship. And so these figures on the right side, we have Pauoa on the top and Kukio on the bottom. We have salinity on the x-axis and then nitrate on the y-axis. And we found that we ran regressions which came out as weak, but they were significant. And comparatively, there is a smaller range for Kukio in terms of salinity versus Pauoa, but it still shows that there is a relationship between SGD and nutrients that are present on the reef. So the next question is, we know that there are nutrients coming through submarine groundwater discharge at these coastal waters, but how long are they there for? Or how long are they bathing the benthos for? And so as we saw with the, that there's higher nutrient concentrations for some of those surface stations, and there's the relationship between the benthic salinity and the benthic nitrates that we looked from the logger data, we were able to show a distribution of salinities. So the figure on the right here shows Paoa on the top and then Kukio on the bottom, with stations on the x-axis and salinity on the y-axis. And then again, it's just the box and whisker. So the min and max is the vertical line, and then the line in the box, the horizontal line in the box, is the median salinity for each of the stations. And so 16 out of the 23 stations were observed to have a low salinity. And so anything less than 33.5, full strength seawater is about 35. The average benthic salinity for both developments was about 34. So anything less than 33.5 was considered having low salinity. And so what we found was that these low salinity events could range from anywhere from minutes to up to 2 days. And that there were 2 stations at both developments. At Paoa, there was 2 stations that were at these low salinity events for 21% of the deployment. And then Kukio also had 2 stations that were at these low salinity events for 23 to 28% of the time. So to sum up my objective 2, it was to just look at the spatial distribution of nutrient concentrations, really looking at the SGD component and how long those nutrients are on the benthos for. And we saw that there was evidence of SGD at both developments, and that more specifically, groundwater wells, shoreline, and surface waters did have stations that had elevated nitrate concentrations, and that benthic salinities and nitrate concentrations did have a relationship, and low salinity events occurred at both of the developments, but with varying durations. So in conclusion, we found nutrient concentrations were higher at the shoreline and at surface waters, and that the use of stable isotopes, nitrogen-15 in macroalgal tissue, as well as the nitrogen-15 in nitrate, using those through mixing models, we were able to successfully identify the nitrogen sources and provide a percent contribution for the sources as well. And this ties in with previous research done in West Hawaii, where the dominant and nitrogen sources were found to be like around high elevation groundwater, some ocean, and then also very heavily influenced by landscaping and suggested by fertilizers. We also found that SGD is a nutrient source to coral reefs. The sensors that we deployed captured these SGD patterns, and that these pulses can vary from minutes to days, and that using this information, we could see that reefs are being exposed to nutrients from land, and that these low salinity events can cross over into the daytime. So this provides more opportunity for algae, which are photosynthetic and can take up more of those excess nutrients during the daytime and start creating a problem and impairing coral reef health. And this is useful for understanding nutrient dynamics on reefs, really finding susceptible areas to nutrient pollution. Some of the implications with this study is that using that dual approach, we were able to successfully identify nitrogen sources, and there was better spatial mapping of nutrients, identifying the location of nutrients and their inputs from a watershed level. So from those groundwater wells and going to inguline ponds, shoreline, and then offshore stations. And that the benthic SG patterns identified locations on the reef that were experiencing longer exposure to nutrients than others. Other implications is from a management perspective is that managers can use the stable isotope mixing model to better pinpoint locations for management actions and overall just minimizing nutrient inputs. So this can be anything from landscaping changes, reducing fertilizers, or removal of nitrogen fixating vegetation or creating riparian barriers. And by doing all of this, you can also create collaborative partnerships with managers, stakeholders in the community to continue monitoring areas with coral reefs and see if there's a change in water quality by minimizing these inputs. And all of this will build resilience for coral reefs by reducing a stressor that can actually be managed at a local scale and kind of reduce that threat or the stress of some of the global threats that I mentioned earlier, like ocean acidification and rising temperatures. And future steps with this project would be long-term monitoring, including other nitrogen sources in other areas, and you really applying these approaches to other locales with coral reefs and SGD as their main input. So looking at other islands as well. So I'd like to thank my advisor and my committee members for supporting me through my thesis. And I would also like to thank all of the wonderful people who made this project possible. The NOAA Habitat Blueprint Office, the NOAA Pacific Island Fisheries Science Center, the Nature Conservancy, the UH Hilo Marine Science Department, the UH Hilo Analytical Lab, UH Manoa, Hawaii Institute of Marine Biology, PIPES and their interns, TCBES, cohort, and everyone who's helped support and make this project possible. Thank you so much. And I'm happy to take any questions.
I have a question.
Yes. Becky? Yes?
Yeah.
Okay.
So you're showing that there's definitely some kind of anthropogenic sources in these sites.
Mm-hmm.
How big of a problem is it? Like, what, based on what you've seen in pollution in other places in Hawaii, what is the magnitude of this relative to other places? Is this low levels, high levels, intermediate levels?
Well, let's see here. Let me bring up.
Can you say?
I think for this study, we can compare it to like the Department of Hawaii of Health standards, which all of the nitrate levels exceeded for the surface waters. But we know that in West Hawaii and Hawaii in general, that there are just general elevated nutrients. And so until there's kind of like a baseline of knowing what those concentrations are, it's difficult to say. Although, because we know that there's kind of that threshold from the Department of Health, I think it's at about 10 or so. Pretty much all of the stations exceeded that, going even into the hundreds. So these warmer colors here are 127 to 201 micromoles per liter. But I think more research and possibly like modeling of these inputs can help identify the greater magnitude of it.
Hi, Jasmine. Can you speak a little bit more to the importance of collaboration that came along with this, especially regarding the communities where you were working?
Yeah. So these were developments that do have a lot of recreational use, and they, you know, they're right on the shoreline. They have corals. Many people use them, and you know, the community is really concerned about seeing those corals decline, because a lot of people have, you know, been in the area or lived there for quite a while, and they've seen, you know, that the corals go from very vibrant and having a lot of fish to being not so vibrant and seeing some bleaching and just seeing that decline. And so having those partnerships with the Nature Conservancy and NOAA and the community really ties together in both the citizen science and research to help improve coral reef health in the long term, and provide the community with what their water quality is looking like and what they can do to improve and reduce their nutrient inputs to the waters.
Thank you.
Jasmine, I was wondering if you could add a little bit more of what groups we worked with in order to be able to accomplish this project.
Mm-hmm. So we worked with the developments, we worked with those who are supplying the drinking water, those who are you know, going through the effluent or the wastewater, and just a lot of, and also the government organizations, so NOAA, and here, I'll go to the last slide. And also including the academic side as well, with partnering both of the universities to really assess and bring together everyone to look at the water quality for these areas.
I've got another question for you. Can you talk a little bit about the difference in the soil values between the two sites? There was a considerable difference in the delta N15 values that you mentioned. What's going on there? Any ideas?
Yeah, so for Paoa, we see that the fertilized soil is kind of a tighter range, and it's more enriched, or it has higher values of the stable isotope. And then for Kukio, it's kind of a wider range, ranging from -7 to about 5. And so this is probably most likely from using dry fertilizers, and using fertilizers will decrease the nitrogen into negative values. Previous literature ranges it from, let's see, where was it? From about -10 to 15. So that's right where this development falls under, a little bit less. So those kind of two extremes, and also a smaller sample size for Kukio for the soils may have also contributed to that wider range, but it's still a lot more negative and less enriched than the soils at Paoa.
Oh, that's interesting. Thank you.
Hi, Jazz. Good job.
Thanks.
I was wondering, did you look at all at the amount of rain over there and or the currents and take that into consideration in looking at your data?
So yeah, another part of the project was they deployed an ADCP, and it was found that Paoa didn't really have a strong directionality, whereas Kukio, I believe, was towards the northeast. So, and there wasn't really, I mean, we sampled during the summer where there was not really any rainfall at all, especially on the leeward side of the island. So there's no real evidence for any kind of surface flow going into the waters at the time we sampled. And I don't think that currents would play a huge role, probably at Kukio, just because everything was more uniform. But it didn't seem very strong for both developments.
Cool. Thank you.
Yeah.
Any more questions for Jazmine before we wrap up the public part of her defense for today? Okay, great. Thank you, everybody, for coming today. We really appreciate your support. So this wraps up the public portion of Jazmine's defense. She'll now be meeting with the committee member to go over some comments. So thank you, everybody, and have a great rest of your day. And we'll see you soon. Thank you.
Yeah, thank you.
Nice work, Jazmine.
Thank you.
You had a great group out there. Good
Yeah.
to see you. Good to see
Great
people
job,
from all
Jazmine.
over. Some people
you.
from the west side too.
Yeah.
You just get Stu here. Okay. Hey, Jasmine, I don't know if you can
Yeah.
see the comments.
Where? On the chat?
The
Yeah,
chat,
make sure to read through the chat.
And there were a couple other people, I never let them in because I had no idea if iPad 2 or my PC were real people, so.
Yeah, probably not.
I have a feeling iPad 2 was not.
Yeah.